Today we will talk about molecular taxonomy. So molecular taxonomy is what? It is the taxonomy which uses molecular characteristics like the macromolecules such as DNA and protein. Particularly DNA and protein, these two macromolecules are used for classifying bacteria. So these molecules are called molecular chronometers. Molecular chronometer. What is the meaning of molecular chronometer? Chronometer is a kind of a clock. It's a clock. Chronometer means clock. Because these particular molecules, DNA and proteins, their sequences, they helps us to know when had the last significant change or evolution took place. Significant mutation that led to a new species took place. It helps us to know that particular time, approximately when this organism, a particular species, is split into two different species because of a certain mutation. So they are like a clock, isn't it? They're like a clock. That's why they are called molecular chronometers. This question is sometimes asked. So what are molecular chronometers? So molecular chronometers are these DNA and proteins. So how do they help in our taxonomic identification or classification? There are many techniques. The first technique that is discussed is a little bit classical, that G plus C moles percent. This thing, G plus C. In a sample of DNA, you will have G, C, A, T, everything, these four nucleotides. And they have a different composition. Each DNA has a different composition of G, C, and AT. A DNA comprises of G, C, AT, isn't it? And each organism, the genome of the organism, will have a different GC and AT composition. Depending on that, you can group organisms. That these organisms, they fall into a certain G plus C percentage group. Another that one falls in another G plus C percent group. Like this, you can group organisms. This G plus C will not help you in speciation, species identification or genus identification. This will only help you to group some organisms together that they belong to G plus C moles percent 40 to 50%, or 60 to 70%. That will be too high. 50 to 60%, like this. So grouping is possible, that you can make certain groups out of this G plus C moles percent. So G plus C moles percent is determined with the help of, there are two techniques. One is temperature based technique to calculate the Tm value of the DNA. Tm value of the DNA. And the another one is the buoyancy. Buoyancy of the DNA. Buoyancy means plobota, floating characteristic. At what force the liquid on which it is floating is exerting on the molecule. More the G plus C content, more is its sedimentation rate. So that also helps in knowing the G plus C percentage of a DNA sample. Tm value is very important. This helps us at various places. Wherever we go to hybridize our DNA with another DNA, we need to know the Tm value of that particular DNA, the probe with which you are hybridizing, the primer with which you are going to do the PCR, polymerase chain reaction. So at what temperature that primer will hybridize with its template DNA, that needs to be known. You have to set the temperature in your machine according to that. Okay, At this temperature, primer annealing will take place. At this temperature, my probe will hybridize with the target DNA. So if I raise the temperature above that, it will not hybridize. If I keep the temperature much below that, it might hybridize to irrelevant sequences, which is not its own sequence, might be a little bit different sequences. So non-specific, that is called non-specific binding will occur. So to avoid that, you have to know the right temperature at which it will bind to specific sequence. Okay. So for that reason, we need to know the Tm value. What is Tm value? Tm value is the melting temperature. It is the melting temperature of the DNA. It is the temperature when we melt a DNA. What happens? The double helix becomes single helix. The DNA becomes separated. The double helix becomes separated because they are binded together with hydrogen bonds that melts. Okay. And GC-rich DNA, a DNA rich in GC, will have more number of hydrogen bonds than a DNA rich in AT. Because you know, A and T, they form 2 hydrogen bonds among them, between them. And GC forms 3 hydrogen bonds. So automatically, a DNA containing more number of GC in the sequence will have a greater number of hydrogen bonds. So you will take a higher temperature to melt those DNA. So there is a direct relationship with the temperature. So Tm value is a temperature at which the DNA is half melted, you can say. The DNA, when it is half melted, when you are raising the temperature, the DNA is getting melted. When it is half melted, that temperature is called the Tm value. In other words, when you are melting the DNA and you are measuring the optical density, the absorbance of the DNA, at what wavelength? 260 nanometer wavelength. At that wavelength, if the absorbance is half the temperature at which particularly, the absorbance is half the maximum, that temperature is called the Tm value. Let me explain you further. So this is a double-stranded DNA, say, for example. When you are raising the temperature, when you are increasing the temperature, then what happens? The DNA gets melted like this. It is getting separated. And ultimately, if you raise the temperature, say this is, you raise to maybe 55 degrees centigrade. At this temperature, this is like that. At 80 degrees centigrade, this has almost, it has all separated almost. Or if you raise to 90 degrees, it will completely get separated like this. Okay. So there is the temperature when it is half melted, isn't it? And how you can know that temperature when it is half melted? By this measuring the absorbance of the DNA. If you measure the absorbance of the DNA when it is double-stranded, it will be less. How do we measure the absorbance? Using spectrophotometer. So here you will need UV spectrophotometer, a spectrophotometer of UV range, because you need to have 260 nanometer wavelength. A spectrophotometer produces a monochromatic light. It's a machine which will convert the white light into a monochromatic light. Monochromatic means having a specific wavelength. So you can produce, with the help of this, you can produce 260 nanometer wavelength of light. Okay, so this light, remember, the DNA absorbs maximum at 260 nanometer. This is another thing to remember. At what wavelength the DNA absorbs maximum? That is 260 nanometer. We always measure a chemical substance through spectrophotometer at a particular wavelength. That wavelength is the lambda max or maximum absorbance wavelength of that substance. Here we are using 260 nanometer. That means it is the wavelength that is absorbed maximum by the DNA. Not other maybe 250, the DNA will absorb less. 270, it will absorb less. 300, 400, it will absorb further less. Okay. So that's why when you pass a 260 nanometer of light through a solution of DNA, it will absorb the light. but it will transmit also to some extent. How it will look like? Say at 40 degree, you got an OD of 0.5. At 50 degree, you got an OD a little bit higher, maybe 0.75. At 60 degree, you got a higher, maybe 1.3 or something like that. At 70 degree, you got even higher at about 2. And after that, at 80 degree, you got a little bit higher, 82. At 90, 82 again. At 100, 82 again. So it has got melted now. So your graph comes like this. It is like this. So what is the maximum absorbance? This is the maximum absorbance. At what temperature that maximum absorbance was reached? At about 80 degrees centigrade. So what is the half of that absorbance? That is, if it is 2, so that will be 1. So here is the half absorbance. At what temperature that is happening? At nearly 57, 55, 57 degrees centigrade. So for you, this DNA sample, whatever arbitrary DNA sample that you have taken, the Tm value is nearly 57 degrees centigrade. Okay, so you have to find out the Tm value. This is how we find the Tm value of a DNA. This is how a Tm value of a DNA is determined. Okay, so once you know the Tm value, you can determine the G plus C moles percent. There is a formula that is used to determine that. So Tm is equal to 69.3 plus 0.41 G plus C percent. So you can put the Tm value here, 57 degree, to determine the G plus C percent of that particular DNA. And for many other samples, the Tm value is not determined like this. If the DNA is a small oligonucleotide, maybe 18, 20, 25, 30 base pair bases long, then you can use this formula. Tm is equal to 2 into A plus T into 4 into G plus C. So you have to see, you know the sequence already. That means you know the sequence. When you know the sequence of a DNA, then you can use this formula to determine. So this is done when we generally do, hybridization or PCR, polymerase chain reaction. Okay? When we do polymerase chain reaction of a particular DNA, to amplify that DNA, we need 2 primers. And these primers are known sequences because the primers are synthesized, chemically synthesized. When we know the template DNA, we synthesize the primer accordingly. Primers are known sequences. So your primer will bind at what temperature with the DNA that you can calculate via this method. Suppose your primers contains A, G, T, C, G, C, T, A, C, G, G, C, T. Say this much. So what will be the Tm of this? You just take a count of this. How many A, T are there? 1, 2, 3, 4, 5. So A plus T, that is 5. 2 into 5, 10. Plus 1, 2, 3, 4, 5, 6, 7, 8. 8, 4 into 8 is the G plus C. So 32, isn't it? 10 plus 32, 42 degrees centigrade will be the Tm value of this DNA sample. So it's very small. It's very small. So the DNA can be much higher. How many of this? 8 were GC and 5 were AT in this DNA sample. Okay, so using this formula, you can calculate the Tm value, and further you can calculate the G plus C moles percent of a DNA. So this is what is Tm value and GC percentage is. Anyway, so the first thing is G plus C moles percent. The second thing, very important, is 16S rDNA sequence analysis. So this is the second point, second thing. So, but actually this is the most important criteria, molecular criteria, by which we usually determine the, at least up to the genus level, we use this 16S rDNA sequencing to know a particular organism, what is the genus of that organism. So nowadays the scientists are not accepting the 16S rDNA sequencing sequence for species identification. They are saying that you have to do DNA-DNA hybridization with the type specimen. But up to genus level, there is no problem using 16S rDNA. Sometimes you can also use this for species identification. It depends. So you don't need to do even the DNA-DNA hybridization. When you know the entire sequence of the organism, you also know the entire sequence of the type specimen. So in the computer, you can match your sequence, your organism's whole genome sequence with that organism's whole genome sequence. But when we are not able to do the whole genome sequencing, we have to depend on DNA hybridization. I will tell you how. Okay? Are you getting these things or not? 16S rDNA sequencing, I have already told about that to some extent, that this 16S rDNA, there are other rDNA like 5S rDNA. Actually, there are RNA, 5S rRNA, 16S rRNA. These rRNAs are present in ribosomes. And there is a respective gene for that is called DNA, 16S rDNA. Okay. In the ribosome, in bacterial system, there are two subunits, 30S and 50S. In the 30S, you will get the 16S rRNA. This is 16S rRNA. In the large 50S subunit, you will get a 23S rRNA, and there is a small RNA that is called 5S rRNA. So all of them can be used. All of them can be used for, the taxonomy, bacterial taxonomy purpose. You can sequence them. But we will not sequence the RNA, we will sequence the DNA, the respective DNA from where these RNA were formed. RNA is synthesized from the DNA. So there will be a place where the sequence for RNAs are there in the DNA. So you need to sequence that. Okay. So once you get the sequence, you can compare it in the data bank, in the database that is present in the internet. You can compare with the existing data bank that with what organism your query sequence is matching. This is called homology searching. You can match the homology of this 16S rDNA with the existing ones. The computer will say, see, your DNA is matching with such and such organisms. 100% with this, 99.9% with this, 90% with this, and 89% with this, like this. So on that basis, when it is matching 100%, that means it is completely the same organism. And when it is matching 99% and above, that means it is approximately the same organism, little bit one or two sequence differences are there, isn't it? So you may, in that case, when it is 99%, then it is not exactly the same species, or it may be the same species, a variant of that, okay. So it depends. So you need to do some other techniques also, that's why. You need to go for the whole genome sequencing. Because your 16S rDNA sequence differs a little bit, maybe one nucleotide. How big is this 16S rDNA? About 1500 bases. I'm saying about a round of 1545 or something like that is there. So the genome of the organism is maybe 10 to the power 6 base pairs, and out of that you are matching only this much. It will not give the right result. However, this 16S rDNA and similar other cytochrome DNA, gene for the cytochrome, similar other genes which are called conserved genes, they help in our identification purpose. Conserved means not 100% conserved. A gene which is 100% conserved and a gene which is 100% variable will not help you in taxonomy. 100% conserved means all of them will be same. There will be no difference. So how do you make these differences? 100% variable means all of them are different. So how do you match the similarity? So there has to be an in-between situation when the conserved gene undergoes mutation at some point of time. When in the course of evolution, it undergoes mutation. That mutation creates a new species. It might create a new species. If it occurs at a specific place, it might create a new species. So you will notice that when that change occurred, that is one thing. And since it is almost 99% similar to the other one, so that particular mutation has actually split that species into two different species. It might have done that. So these kind of conserved genes help you to match with a particular existing sequence and to determine to which organism it belongs. You can also use the 5S rRNA, 5S rDNA or 23S rDNA, but we do not use that. Why? Because the 5S rDNA is too small and the 23S is actually too big. So in between situation, 15S rDNA is good enough. Okay, but nowadays what I said, that whole genome sequencing has become easy and less costlier, so people are going for whole genome sequencing randomly. Okay, so now let's talk about the whole genome sequencing. What is it? Sequencing the entire genome of an organism. You know, genome means the total DNA of an organism. Om, O-M-E, it sounds like that our Om, entire Brahman. So here also entire DNA. There is similarity with that. Proteome, there is another word. There are this Om word, genome, Om suffix. It is being used at different places, like proteome, transcriptome, metabolome. Okay, similarly lipidome. So all of them speak about the totality, study in totality. Study of total protein of an organism is proteome. Study will be called proteomics. The study will, if you call it study, then I will call genomics. Genomics research, genomics study. That studies about the whole genome. Genome means the whole genome, I mean the whole DNA. Proteome means the total DNA of that organism. The study will be called proteomics. Okay, this will become mix. Proteomics, transcriptomics. Study of the total transcript. The total transcript is called transcriptome. Study of that is called transcriptomics. Study of the total metabolite. Our total metabolite, that is metabolome, it's called metabolomics. Metabolomics. And lipidomics means study of the total lipid, similarly. So you can use OME anywhere, you can put as a suffix. It will make a sense that you are studying in totality of that particular. So anyway, all these things have come up recently. Whole genome sequencing, it started sometime during 1990s, okay, with the Human Genome Project, you can say. And there were many bacterial genome projects. The first bacteria that was sequenced was Haemophilus influenzae, I remember. Haemophilus influenzae, that bacterium, that is not virus. Influenza is caused by bacteria also, Haemophilus influenzae. After that, during that time, when was that sequencing done? 1995 or something like that, 1995. And that was the period when whole genome sequencing was going on randomly on different organisms. On one side, on eukaryotic system like human genome, Drosophila genome, rice genome, Leishmania. In our lab, we used to do Leishmania genome sequencing. In our lab, it was in a collaboration with the French lab. Christine Blano was the scientist I remember, with which my guide, Dr. Addo, he had a collaborative work, and our lab was given a task to sequence the chromosome number 10 only. In Leishmania, there are 36 linkage groups, 36 chromosomes. So a little bit more difficult than human genome project, actually. So that was a period when people used to do sequencing using Sanger's dideoxy method. That is a little bit slow method. During 2005 and afterward, new next generation sequencing, that is called next generation sequencing, that evolved. Now we are running with the fourth next generation sequencing, in the fourth generation of NGS. That is called NGS. Sequencing very fast. There are many techniques of next generation sequencing. Here we will not talk about that. It will need maybe 2-3 different classes, and this is not the place where I should talk. You just know that NGS is the next generation sequencing in which Sanger's method is not used. Pyrosequencing was the method that was being used at the beginning, but there are other methods like people use. There is a system called PacBio system. There is a system in which there is a that is called nanopore system. There is a protein which has a nanopore through which the DNA passes, and there is a fluorescent light that strikes the DNA one by one, and it determines what nucleotide passed through that. So that is the fastest method that has come up, actually. So these methods actually have speed up the thing maybe to a greater scale, I mean mega scale, 10 to the power 6 or 10 to the power 9 giga scale level, they have increased. The speed of sequencing. Using Sanger's DNA method, it was very slow. I remember there is a Sanger's Institute. We used to talk about when human genome project was going on. So the governments, they implanted many sequencers in a particular institute. The whole institute was filled with sequencers that were engaged in sequencing the whole genome sequence of human being. And it took about 13 years to complete the process. Nowadays, you can complete it in 1 week. Okay, so there are things like that. So when you do whole genome sequence of an organism, you have the sequence of each and every part of the genome. So now your thing becomes much easier, or you are knowing the whole thing at a glance. You can compare this with the existing data, with the type specimen directly. With the type specimen, your 16S rDNA sequence is also there. In the whole genome sequence, your 16S rDNA sequence is there, your cytochrome sequence is there. All genes are there within that. The non-genic areas also. Area which are not genes. Everything is there in the whole genome sequence. So now you can compare taking the whole genome, or you may take specific genes to compare with the database. There is a database in your mobile also. You can go and you can search. You can write in the Google BLAST. Just do it now. You write BLAST. BLAST. This is the full form of this is what? Basic Local Alignment Search Tool. Okay. So whole genome sequencing, if you get a whole genome sequence, you can compare the whole genome with the whole genome. Isn't it? So जो भी हो, तो next इससे पहले when whole genome sequence was not available, so then this was the technique, DNA hybridization. You can use this even today also. This is comparatively much easier. DNA with DNA. Duto DNA. Which DNA with which DNA? One is your DNA, query DNA, the organism that you have isolated, you have sequenced. Either you have done 16S rDNA sequencing, you have not done whole genome sequencing. That is not available to you. But this thing is available. Okay. So you have got the sequence of you have got the DNA type specimen. You have got the type specimen. And this DNA is from the query DNA, query organism that you have isolated, and that is new one. You want to know what organism is this. For that purpose, we are doing taxonomy. है ना? So type specimen is what? This is that specimen on the basis of which the name of the organism was given. The name of the organism is associated with that specimen. That is the type specimen. Where from you will get the specimen? You will get the specimen from certain bank. Certain organism banks are there. Culture collection centers like the ATCC, American Type Culture Collection Center. This is one of the largest, I think the largest one. ATCC. In India, you will get maybe MTCC, Microbial Type Culture Collection. Microbial Type Culture Collection. It is at IMTech, Chandigarh. And there are other places also. Good enough. You might get your type specimen from these places. And then you can isolate the whole DNA from there. You will isolate the whole DNA of your query organism. Okay? Now you have got two DNA samples. One is of query and one is for the type specimen. We can identify, we can match our organism's DNA with a type specimen DNA. ঠিক আছে? Then we can, আর আমি বলবো এখনই না বলি. There is called one technique called ribotyping. Ribotyping, there is this technique. This technique uses a genetic engineering method called, molecular biology method called RFLP, restriction fragment length polymorphism. This uses a technique called RFLP, restriction fragment length polymorphism. इसमें क्या होता है ना कि हमको टाइप करना है राइबो माने RDNA को टाइपिंग करना है. ओके, RDNA के फाइंड आउट করতে চাইছি. আমার কাছে হোল জিনোম আছে, এটা ব্যাকটেরিয়াটা আমার. This is my bacterium. This is the genome of the bacterium. You isolated the genome. You digested with certain restriction endonuclease. Suppose, for example, eco R1. So you got several fragments of that. It got fragmented, isn't it? Some of the fragments have RDNA also. A total genome fragment. ওর মধ্যে রাইবোজোমাল ডিএনএও থাকবে, আর 16S RDNA, 23S RDNA গুলো থাকবে. এবারে you run a gel. After this you run a gel. Gel run করলাম, ইলেকট্রোফোরেসিস করলাম. ওকে, তো জেল রান করব, তো এগুলো হচ্ছে ওয়েলস যেখানে ডিএনএটা লোড করা হয়, আর ইলেকট্রিক কারেন্ট, এদিকে নেগেটিভ চার্জ এদিকে পজিটিভ চার্জ, ইলেকট্রিক কারেন্ট সাপ্লাই করা হয়, তখন এরা ব্যান্ড ফর্ম করে এদিকে মাইগ্রেট করতে শুরু করে. ডিএনএ নেগেটিভলি চার্জ ও নেগেটিভ থেকে পজিটিভ চার্জের দিকে যেতে শুরু করবে. অ্যান্ড দিস ইজ মেড আপ অফ অ্যাগারোস, সো দে উইল মুভ উইদিন দি অ্যাগারোস. So this is called agarose gel electrophoresis. So when you do electrophoresis, what will happen? These bands will get separated. Ora erokom bhabe ekhane khub closely they will get separated like this, because this is whole genome. You will get a sequence like this. And not sequence, I mean a picture like this in the gel. Because this is a whole genome, and it has got all types of fragments, from very small to very large, a continuous size of fragments. So there will be a smear. This is smear of DNA. यहाँ पर कोई शार्प बैंड नहीं दिख रहा, तुमको स्मीयर दिख रहा है, ठीक है? इसी तरह तुम्हारा अननोन बैक्टीरिया, सपोज दिस इज अननोन बैक्टीरिया, द बैक्टीरिया क्वेरी बैक्टीरिया, क्यू लिख सकते हैं इसको, क्यू क्वेरी बैक्टीरिया. बट you have doubted, not only doubted, you have done some experiments like those phenotypic tests, biochemical tests, by which you have almost reached to the genus level. Maybe you have done 16S RDNA also, so very much you also know approximately the species also. Okay, now you are doing this thing, so you know this is similar to Bacillus subtilis. So you take Bacillus subtilis, Bacillus stereothermophilus, Bacillus megatherium. So you took several species of bacterium, their DNA, and you did the same thing. You isolated their DNA, you digested with EcoR1, and you run in the gel with this one. Parallelly to this one, your unknown one, you run in the other lens, those DNA were loaded. So they will also form bands like this. Why I am drawing like this? Because total genome ke jokhon restriction digestion korbe na, to sob rokom fragment produce hobe, sharp band pabe na, etake bole smear. ऐसा स्मीयर आएगा. नाउ अब इसको हम हाइब्रिडाइज करेंगे किस चीज से? राइबोसोमल डीएनए, जो भी हम 16S RDNA ले सकते हैं उसको प्रोब बना लेंगे. 16S RDNA you will use that as a probe, and you will hybridize this particular gel. पहले जेल से इसको हम लोग एक, देखो यहाँ पर सब कुछ बोलना पड़ जा रहा है, तुम लोग स्किप कर जाना, बताने के लिए बता रहे हैं, समझ में नहीं आ रहा बाद में आ जाएगा समझ में, ठीक है? लेकिन हमको तो स्टोरी थोड़ा बताना पड़ेगा, ऐसे पूरा जम मारेंगे तो तुमको लगेगा क्या हो गया, कैसे हो गया है? এই জেলটা রান করার পর হাইব্রিডাইজেশন জেলে করা যায় না, এটা থেকে একটা মেমব্রেনে ট্রান্সফার করে নিতে হয় ডিএনএটাকে, কারণ জেলটা ভেঙে যাবে, জেলটা একটু ব্রিটেল হয়, ভেঙে যাবে। তো জেল থেকে ডিএনএটাকে লিফট করে নেওয়া হয় একটা মেমব্রেনে, মানে ওই জেলটাকে এরকম একটা ট্রেতে, ট্রের একটা বড় বড় ট্রে থাকে, ছোটতেও হতে পারে, এখানে গ্লাস প্লেট রাখলাম, এর উপরে ব্লটিং পেপারের একটা ইয়ে ঝুলিয়ে দিলাম দুদিকে, ঠিক আছে? এটা ধর খোলা আছে, হ্যাঁ, এখানে একটা গ্লাস প্লেট রাখলাম, গ্লাস প্লেটটা ছিটিয়ে দিলাম সেলো টেপ দিয়ে, দিয়ে একটা ব্লটিং পেপারের স্ট্রিপ এরকম ঝুলিয়ে এর মধ্যে ঢুকিয়ে দিলাম, এর মধ্যে কিছু বাফার ঢালা হবে, ওই সোডিয়াম ক্লোরাইড, সোডিয়াম সিট্রেট, ওর উপরে নির্ভর করে ওই বাফার, ওটা এসএসসি বাফার বলে, ওটা দেবো, ওর মধ্যে ব্লটিং पेपरগুলো ডুবে যাবে, পুরো ভিজে যাবে। उसके ऊपर एगरोज का जेल, उसके ऊपर एक मेम्ब्रेन, जो जिस मेम्ब्रेन में ट्रांसफर करेंगे, उसके ऊपर फिर ब्लॉटिंग पेपर, उसके ऊपर ब्लॉटिंग पेपर का एक इतना मोटा लेयर, काफ़ी सारा ब्लॉटिंग पेपर, फिर उसके ऊपर एक ग्लास प्लेट और उस पर एक मोटा सा किताब रख देंगे, रात भर छोड़ देंगे। इससे क्या होगा, वो धीरे धीरे ब्लॉटिंग पेपर भीजता जाएगा ऊपर वाला, और नीचे से डीएनए को ऊपर की तरफ कैपिलरी एक्शन से ऊपर ले आएगा, और वो मेम्ब्रेन के साथ वो डीएनए चिपक जाएगा, अटक जाएगा। मेम्ब्रेन जो है वो पॉजिटिवली चार्ज है, तो डीएनए को अट्रैक्ट भी करता है उसके साथ वो बाइंड करने में। उसके बाद एक क्रॉस लिंकिंग मैकेनिज्म है, वो हम डिटेल्स में अभी नहीं बता रहे हैं, तो मतलब उस पर लग जाएगा। अब मेरा जो डीएनए है वो मेम्ब्रेन में आ गया है, दिस इज नॉट इन द जेल नाउ। সো এবারে তোমার এই মেমব্রেনে ডিএনএটা আছে, এইখানে তুমি এরকম ব্যান্ড দেখতে পাবে না মেমব্রেনে, জেলেতে দেখতে পেয়েছিলে, মেমব্রেনে পাবে না। এইখানে হাইব্রিডাইজ করব, একটা প্লাস্টিক ব্যাগের মধ্যে এটাকে নেব, যার মধ্যে হাইব্রিডাইজেশন বাফার থাকবে, আর এর মধ্যে আমি প্রোবটা অ্যাড করে দেব। প্রোব, এই প্রোবটা কি? আরডিএনএ, রেডিও লেভেলড আরডিএনএ। প্রোব বলতে কি? রেডিও লেভেলড। যখনই বলবে প্রোব বা রেডিও লেভেলড কথাটা, whenever I or anyone says radio level, you think about যে it is a radioisotope maybe of, in case of molecular biology, P32. When we are talking about DNA, P32. When we are talking about protein, it is S35 methionine. When you are talking about certain biochemical pathway, it is C14. In one case, it is N15. That is not radioisotope, stable isotope though. That is the semi-conservative model of DNA replication that you study, N15. That one, another thing is this. So, radio level probe. This probe will go and will bind. এই ডিএনএ গুলো এখানে আছে, এর মধ্যে আছে, দেখতে পাচ্ছি না, আছে। ওই দেখার জন্যই প্রোবটা ইউজ করলাম। ওই প্রোবটা গিয়ে কি করবে, যেখানে ওর সাথে ম্যাচ করছে এটা সিঙ্গেল স্ট্র্যান্ডেড ডিএনএ, এগুলোকেও সিঙ্গেল স্ট্র্যান্ডেড করে নেওয়া হয়েছে একটা केमिकल सॉल्यूशन में फैले ওগুলোকে we denatured that। ओ तुम बंगला, हमको बीच बीच में बंगला, बंगाल में है ना, बंगला बोलना पड़ेगा। I have to make people understand by speaking Bengali only, sometime Hindi। So what will happen, your probe goes and binds maybe some, यहाँ bind कर गया, इस कलर से दिखा रहा है, ब्लैक से दिखा रहा है, दोनों में दिखेगा, यहाँ बाइंड कर गया, इधर कहीं बाइंड कर गया, इधर कहीं बाइंड कर गया, ठीक है, मतलब यहाँ पर, यहाँ पर भी है ना वो डीएनए है यहाँ पर, और इसमें से सब उल्टा पुल्टा बाइंड किया। इसमें जो 3 लेन है, one bound here, another here, but this bound here and here, and this bound here and here, suppose। So this is the picture, this is the probe that had bound, but after that binding what you have to do, x-ray film में expose করতে হয় এটাকে, x-ray film এ রেজাল্টটা দেখতে পাওয়া যায়। যেখানে যেখানে রেডিও অ্যাক্টিভিটি গুলো আছে, প্রোব আছে মানে রেডিও অ্যাক্টিভিটি আছে, এবারে এই this membrane has to be exposed to an x-ray film। The technique is called autoradiography। So when you do an autoradiography, your autoradiogram will show bands, black bands on the x-ray film। Those are in the same position where the probe is there, and probe has bound to which place? Its complementary DNA। So this complementary DNA were here, the rDNA in the genome were there। Now you can see that this lane is matching with this lane, the bending pattern matches with particular lane। This is query, and these are different, Bacillus subtilis, Bacillus stereothermophilus, and Bacillus megaterium, these were 3 others। So with which one your query sequence matches, query bands matches, banding pattern, this is the RFLP data, restriction fragment length polymorphism data। Basically it's RFLP that we will talk later, but this is what is called ribotyping। When you do this, with which particular strain or species, which particular lane your query sequence is matching? Ball, third one। That means my organism is the third one, I mean the Bacillus stereothermophilus, not this one, not that one। इसके साथ मैच नहीं कर रहा, इसके साथ मैच नहीं कर रहा, इसके साथ मैच कर रहा है ना, देखो यहाँ पर भी बैंड है, यहाँ पर बैंड है, यहाँ पर बैंड है, यहाँ बैंड है, यहाँ बैंड है, यहाँ बैंड है। अब इसको एक्यूरेटली मेजर करने के लिए तुम इसको जेलडॉक, एक मशीन होता है, उसमें डालकर डिस्टेंस मेजर कर सकते हो कि ऊपर से नीचे लेन का कितना दूरी दोनों का बराबर है कि नहीं, है कि नहीं, वो कर सकते हो, स्केल से माप के कर सकते हो। तो you got in, this is called ribotyping। From here also you get an idea that your species is similar to this one। So single-handedly this technique will not help you, but of course you can start with this also। I mean you have done the phenotypic studies by biochemical tests, you have reached to the genus level, now you are guessing that this could be the species। So on the basis of the guessing, if you have those organisms in your lab or from somewhere you can get it, you can do this। This is comparatively easy than going for 16S rDNA, hard work, but most of the laboratory have this facility, molecular biology laboratory, or you can directly go for 16S rDNA sequencing। Along with that you have also done this, so it is matching with the same, then you are confirmed, okay, that means I'm going in the right direction, isn't it? আচ্ছা, এই টেকনিকটারই আরেকটা অল্টারনেটিভ টেকনিক আছে, ওটাকে বলে আরড্রা, আরড্রা, amplified rDNA restriction analysis। This is restriction analysis, restriction digestion analysis, and another thing is called ARDRA, amplified rDNA restriction analysis। All these things you will get in this book, okay। এই বইটাতে পেয়ে যাবি, এখানে পলিফেজিক অ্যাপ্রোচ বলে দেওয়া আছে, क्राइटेरिया फॉर बैक्टीरियल क्लासिफिकेशन तो पॉलीफेजिक अप्रोच फेनोटाइपिक केमोटाइपिक लेखालाम জেনোটাইপিক और মলিকুলার অ্যাপ্রোচ এগুলো এখানে পেয়ে যাবে এই যে সব ছবি টবি দিয়ে অনেক কিছু বোঝানো আছে हाँ राइबोटाइपिंग आर्ड्रा ये सब समझाया हुआ है यहाँ से तुम लोग कंसल्ट करना है ठीक है तो हाँ आर्ड्रा में क्या है कुछ ऐसा ही है आर्ड्रा में पहले तो हमको एम्प्लीफाइड आरडीएनए पहले हमको एम्प्लीफाइड फर्स्ट यू हैव टू एम्प्लीफाई द राइबोसोमल डीएनए राइबोसोमल डीएनए के एम्प्लीफाई करते हैं you don't need to do the restriction dilution of the whole genome after isolating the whole genome go for PCR amplification means PCR is actually for amplifying the DNA RT-PCR in case of coronavirus people were doing RT-PCR isn't it because the coronavirus has the RNA not DNA so that has to be converted first to DNA that's why reverse transcription the first reaction will be reverse transcription to convert the RNA to DNA and then followed by PCR, polymerase chain reaction, to amplify that particular DNA, which has been converted from RNA to DNA. If your blood sample contains the virus, amplification will occur. If it does not contain, amplification will not occur. Okay? On that was the thing, that was the technique. And how much infection you have, if infection is there, amplification will occur. But how much infection you have got, how do you quantitate? It is the threshold value, CT value that they were calculating. In how many cycles you are getting enough amplification. Okay. In this case, they were measuring with the help of the machine that is also called RT, that is real-time PCR machine. Nowadays, a machine has come, not nowadays, quite older. That is called real-time PCR. It gives real-time information that your DNA is amplified or not. Earlier, we only had the PCR, simple PCR. I have a PCR machine downstairs. But that is a simple PCR. That is not real-time PCR. In real-time PCR, the PCR machine is associated with a spectrophotometer. The machine that we talked about, spectrophotometer, a kind of spectrophotometer is also there in the PCR machine. It's a combination of spectrophotometer and PCR. And you have added, in the reaction, you have added a dye, cyber green. There is a dye called cyber green. This dye has a characteristic that it binds with double-stranded DNA only. So when you are doing PCR, you need to melt the DNA first. Then you add the primers. Primer will go and hybridize here and here. And then it will amplify, it will make double-stranded DNA again. So once the double-stranded DNA is made, PCR में क्या होता है? PCR में छोटा सा समझाता हूँ, ये double-stranded DNA था, इसको PCR करना है. PCR करने के लिए तुमको प्राइमर जानना पड़ेगा, मालूम होना पड़ेगा. इसीलिए जीनोम सीक्वेंसिंग पहले से तो था, कोरोना वायरस वाला तो था ही. और दूसरा केस में तुमको जानना पड़ेगा कि वो सीक्वेंस प्राइमर्स क्या होगा. PCR करूंगा, प्राइमर्स क्या होगा मेरा? A pair of primers you have to use, है ना? तो आपने यहाँ का एक प्राइमर लिया, इधर का एक प्राइमर ले लिया, ठीक है? अब इसको क्या किया? टेम्पलेट डीएनए प्लस प्राइमर प्लस टैग पॉलीमरेज प्लस डीएनटीपीज जो है प्लस साइबर ग्रीन ये सब मिला दिया और पीसीआर मशीन पर सेट कर दिया. पीसीआर मशीन में 3 टेम्परेचर होता है, 3 टेम्परेचर का साइकिल रन करता है. पहला टेम्परेचर है डीएनए को मेल्ट करेगा 94 डिग्री सेंटीग्रेड. दूसरा टेम्परेचर है प्राइमर एनिल करवाएगा, प्राइमर एनिलिंग टेम्परेचर दैट विल बी नियर टीएम वैल्यू. दैट्स व्हाई वी नीड टू नो द टीएम वैल्यू. टीएम वैल्यू থেকে 5 डिग्री कम. देन 72 डिग्री सेंटीग्रेड. 72 degree centigrade is the polymerization temperature because Taq polymerase, you have heard about that, Thermus aquaticus polymerase. This is thermal resistance polymerase. Because যখন তুমি 94 degree করছো to melt the DNA, your polymerase gets degraded. So you need to have a resistant, thermal resistant polymerase. That's why you use Taq polymerase. Adding all these things, you set the temperature, you run the PCR. What will happen? In the first round, it will form 2 strands. In the second round, it will form like this. So every round it will double up. So your fluorescent dye, the cyber green, it binds with double-stranded DNA. And when it binds with double-stranded DNA, it produces a fluorescence. Your machine detects how much fluorescence is emitted, है ना? तो जब वो जैसे जैसे डबल स्ट्रैंड का अमाउंट बढ़ रहा है, फ्लोरेसेंस भी बढ़ रहा है ना? जब बाइंड करते तभी फ्लोरेसेंस प्रोड्यूस करता है. तो कितना साइकिल के बाद ये फ्लोरेसेंस तुमको डिटेक्टेबल अमाउंट हो गया? अगर इनिशियली ज्यादा रहेगा, अगर वायरस का लोड ज्यादा रहेगा तो ये साइकिल्स बहुत जल्दी रीच कर जाएगा. सपोज किसी के अंदर 50 वायरस है, किसी के अंदर 1000 वायरस है, ये वाला 20 साइकिल के बाद हमको जितना जो अमाउंट एम्पलीफाइड होगा, ये वाला 8-10 साइकिल में ही हो जाना चाहिए, 10 साइकिल के अंदर हो जाना चाहिए. दूसरा चीज, स्टार्टिंग मटेरियल যদি 50টি থাকে, স্টার্টিং ম্যাটেরিয়াল যদি 100টা থাকে কি ডিএনএ, তাহলে 100টাকে আমি ওইভাবে অ্যামপ্লিফাই করছি আর 50টাকে ওইভাবে অ্যামপ্লিফাই করছি. দুটো সমান সমান হতে এর কত সাইকেল লাগবে? খুব তাড়াতাড়ি হয়ে যাবে অফকোর্স, और एयर অনেক বেশি সাইকেল লাগবে, है ना? इसी को सीटी वैल्यू थ्रेशोल्ड साइकिल बोलते हैं. इससे समझ में आता है किसी का अगर सीटी वैल्यू 25-30 है, माने उसका इन्फेक्शन बहुत कम है. किसी का सीटी वैल्यू 10-12 है, यानी इन्फेक्शन बहुत ज्यादा है. एनीवे, हम इन्फेक्शन कुछ समझा दिए उधर का चीज. पीसीआर एक्चुअली ये है. पीसीआर भूलना मत तुम लोग, बार बार पीसीआर के बाद कहीं ना कहीं से आ ही जाता है. तो पीसीआर में 2 प्राइमर लगता है. क्यों लगता है? इसको घर में थोड़ा सा रेप्लीकेशन पढ़ना, पता चल जाएगा. और पीसीआर में ये सब एंजाइम ऐसे लगता है, डीएनए सिंथेसिस करना है तो डीएनटीपी सप्लाई करना होगा, एक पॉलीमरेज देना होगा, 2 प्राइमर देना होगा, और पीसीआर मशीन चाहिए. पीसीआर मशीन में हमको 3 टेम्परेचर सेट कर देना होगा. काम मेरा शुरू हो जाएगा. अब हम साइकिल्स रन करेंगे. कितना साइकिल रन करेंगे ये डिपेंड करता है मेरा स्टार्टिंग मटेरियल कितना है इस पे. एनीवे, तो यहाँ पर हमने पहले पीसीआर किया. पीसीआर मतलब हमने ये जीनोम था ना, ये जीनोम आइसोलेट किया था, यहाँ से पीसीआर कर लिया. तो मेरे पास अभी बहुत सारा पीसीआर का. किसका पीसीआर किया? आरडीएनए का, सिर्फ आरडीएनए का. नॉट, हमने आरडीएनए को अलग कर लिया एम्पलीफाई करके. टोटल जीनोम में से आरडीएनए को एम्पलीफाई कर लिया बहुत ज्यादा. अब उसका रेस्ट्रिक्शन डाइजेशन किया. टू डिड अ रेस्ट्रिक्शन डाइजेशन टू दिस. मे बी आप इसको, अब वहां पर इको R1 यूज किए थे, यहाँ चलो दूसरा यूज कर लेते हैं, बैम H1, एक दूसरा रेस्ट्रिक्शन एंडोन्यूक्लियस. उससे कट किया. इससे कट करने के बाद जेल रन किया. जेल में आपको ठीक पैरेलली आपने एक क्वेरी सीक्वेंस, और यहाँ पर उसी तरह आपने बैसिलस सबटिलिस, बैसिलस स्टीरियोथर्मोफिलस, बैसिलस मेगाटेरियम और व्हाटएवर आपने ऑर्गेनिजम्स का भी पीसीआर किया, आरडीएनए एम्पलीफाई किया, and they did a restriction digestion with that also. Now in this you are getting bands, separate bands. Suppose आपको ऐसा बैंड मिला, क्योंकि आपके पास तो स्पेसिफिक डीएनए साइज का डीएनए, होल जीनोम नहीं है. होल जीनोम विल गेट स्मीयर, এই ক্ষেত্রে স্মিয়ার পাবে না, স্পেসিফিক ব্যান্ড পাবে. আচ্ছা, এর এইখানে এসছে, এর আবার যেহেতু সেম ছিল আমি এখানেও সেম রাখছি, এর এইখানে এসছে. So after this you don't need to go for probing, hybridization or autoradiography. Your gel picture will reveal the data. So it is a little bit less cumbersome. Only thing is this, that you have to do a PCR before going to this. So you can do this in lab. In our lab we can do this. We can do this part easily in our lab here. We can do the PCR. Up to that we got the PCR amplified, and then we can go for restriction digestion. That facility is there. So instead of going for sequencing, you can go for sequencing at this level, Cisnus RDNA sequencing. You have to send it somewhere because you cannot do sequencing here. But you can, before going to sequencing, you can do this one when you are screening a lot of bacteria. So you don't need to go for every strain for sequencing. You need to select some of them, then you will go, because it is costlier affair. So definitely you have to decide like that. So this is called ARDRA. Now I have already told about FAME technique, fatty acid methyl ester. Okay, what is done in that case? That you can discuss under this also in molecular. criteria. Though that is in chemotaxonomy, but you can transfer that from here, there to here in FAME. That is called FAME technology. Fatty acid methyl ester. Here you have to extract the fatty acid from the bacteria. The whole fatty acid extraction. And then the fatty acids have to be converted to their methyl derivatives. First you isolate the fatty acid of a bacterium. Fatty acid. Go for methylation. So they will become methylated derivatives of fatty acids. Why do you methylate? To make them volatile. Because fatty acids cannot be subjected to gas chromatography. So after that you have to go for gas chromatography, GC. But you cannot directly go to GC with the fatty acids because they're non-volatile. Gas chromatography needs the substance to be converted into gas. It has to be in a volatile form. So for that purpose, this methylation was done. Now you go for GC, you will get a result somewhat like this. There will be many curves like this. This is gas chromatogram, and you have to compare this chromatogram with the database. Because these peaks are actually for some fatty acid. All of these peaks they represent some of the fatty acids. So what is the composition of the fatty acid of a particular bacterium? You can determine by this method. This will also help you to match with the known organism, similar organism, to reach up to maybe species level. It will help you along with the 16 rDNA sequence analysis. Okay? এইটা হলো FAME. যাই হোক, তো এগুলো যথেষ্ট now, not more than this. I think this will be more than enough. Molecular taxonomy. You can watch my video. There are previous videos also. Mol tax নামে দুটো ভিডিও আছে, দেখতে পারিস।